Hi friends, welcome to my channel. We are studying dark reaction of photosynthesis. So we are watching part 2 of the video. In the last class we have seen part 1. Let us watch part 2 of dark reaction of photosynthesis. So we have seen in the last video what all we have seen. Please revise that and then check the second video. So in the last class we have seen the structure inside the chloroplast, what is happening inside the chloroplast. So we have seen in photophosphorylation, that is addition of phosphate, and oxidative phosphorylation, and that is also making of ATP. For ATP synthesis, ATP synthesis uses chemiosmotic hypothesis. Chemiosmotic hypothesis. So let us revise. This is a chloroplast and this is the outer membrane and this is the inner membrane of chloroplast. Chloroplast is a double membrane structure. This is the outer membrane, inner membrane, and these are the granule thylakoids. Thylakoids, they are arranged as stacks one above the other. So this all together is known as the granule thylakoid. Now the membrane of chloroplast, it is made up of lipid. It is a lipid bilayer. Even the membrane of granule thylakoid, the membrane of granule thylakoid, it is also made up of lipid. It is a lipid bilayer. Now attached to this membrane of granule thylakoid, there is a transmembrane channel. This is the transmembrane channel, and this transmembrane channel is made up of two things. First one is F1. F1 is the main channel. Through this channel, the protons, they come out. Through this channel, the protons, they come out. F0 is the channel and this is the F1 is the head. So this is a transmembrane channel. F0 is the channel and F1 is the head. It is a transmembrane channel is attached to the all over the granule thylakoid, so that the protons, they can come out to only through this channel. And for chemiosmotic hypothesis, we have studied that the concentration of proton should be more inside the lumen of the thylakoid. This is the stroma, matrix of the thylakoid, than the stroma. In the stroma, if the protons concentration is 100, then inside the lumen the number of the protons should be 200. So for chemiosmotic hypothesis or for ATP synthesis, 4 conditions should be satisfied. 4 conditions are required for ATP synthesis. Now let us study the 4 conditions. Before that, once again we can revise the structure of chloroplast. The thing is, inside the granule thylakoids, the number of protons should be more than the number of protons in the stroma. More protons should be accumulated inside the lumen of the granule thylakoid, then only ATP synthesis will take place. So for ATP synthesis, 4 requirements or 4 conditions are there. The 4 requirements are, for ATP synthesis, 4 requirements are, first one is proton gradient. What is proton gradient? Concentration difference of protons. Proton gradient is concentration difference of proton. So proton gradient, it should be more inside the lumen of the granule thylakoid than the stroma. So the number of protons should be more inside the lumen of the granule thylakoid than that in the stroma. So more protons should accumulate inside the granule thylakoid. So first one is ATP. For ATP synthesis, there are 4 requirements. For chemiosmotic hypothesis or ATP synthesis, 4 requirements are there. First one is proton gradient or concentration difference in the protons. The number of protons should be more inside the lumen of the granule thylakoid, then only they will move out, and then the stroma. So then only ATP synthesis will take place. Now let us see the second requirement for ATP synthesis. Now for ATP synthesis, we have seen the first requirement. First requirement is proton gradient. The number of protons should be more inside the lumen of the thylakoid membrane when compared to stroma. Now second requirement is intact membrane. The membrane should be intact. That is, protons should not leak out of this membrane. I have told you this membrane is also made of lipid bilayer, and the protons should not leak out of the thylakoid membrane. and the proton should remain intact. The membrane should be intact, so the proton stays inside, and the proton should move only through the F0 F1 channel. The proton should go out only through the F0 F1 channel, and it should not go out through the membrane of the thylakoid, granule thylakoid. The membrane of granule thylakoid, it should be intact, so that the protons should not do not simply move out of the granule thylakoid. The protons, they should move only out of the F0 F1 channel. Okay, so second condition or second requirement for ATP synthesis is intact membrane. The third requirement for ATP synthesis is a proton pump. So there is a proton pump, that is, the proton should always enter inside the lumen of the thylakoid membrane. If more protons enter inside the thylakoid membrane, then only they will move out of the F0 F1 channel, and then only ATP is synthesized. So more protons, they should come out, they should come in, they should go inside the thylakoid membrane, inside the lumen of the thylakoid, then only the protons, they will come out through the F0 F1 channel, and ATP is synthesized. So the number of protons going inside the thylakoid, it should be granule thylakoid, it should be more. Then only these protons will come out through the F0 F1 channel and synthesize ATP. So the third condition is proton pump. More number of hydrogen ions or protons should enter inside the thylakoid, granule thylakoid, then only they will come out through the F0 F1 channel, and they will make ATP. They will help in the synthesis of ATP. So if this is the granule thylakoid and this is the channel, F0 is the channel, F1 is the head. When the hydrogen ions, that is the protons, when they come out through the channel, this head, it will spin or rotate. This rotation of head, it is known as conformational change. This rotation, when the protons, they come out through the F0, this head, it will spin. F1, it will spin or rotate, and this rotation is known as conformational change. The energy of the conformational change, it will help in the combining ADP with phosphate, and hence ATP is synthesized. So once again I will tell you, this transmembrane or this F0 F1 channel is attached to the thylakoid, membrane of the thylakoid. So the number of protons should be more inside the thylakoid, then the protons, they will come out through the F0 F1 channel. So this is F0 is the channel. When the protons, they come out, the F1, that is head, it will spin. So this spinning or rotating of F1 head, it is known as conformational change. This conformational change will combine ADP with phosphate, and hence ATP is synthesized. So we have seen the three requirements for ATP synthesis. Let's pass on to the fourth requirement for ATP synthesis is F0 F1 particle. F0 F1 particle is a transmembrane channel through which the protons, they come out. So this is the requirement, and in the chloroplast, it is known as CF0 F1. In the plant's chloroplast, it's present, so it is known as CF0 F1, and in the mitochondria in animals, it is known as F0 F1. In mitochondria also ATP synthesis takes place, which is known as oxidative phosphorylation. In chloroplast, it is known as photophosphorylation. So the fourth condition for ATP synthesis is F0 F1 particle. So I hope that you like my videos, and I will be covering all the topics for NEET biology for free. Please subscribe, like and share my channel.